Welcome to the deep dive. Today, we're heading somewhere unexpected. From the extreme cold of glacial ice right into the cutting edge of neuroscience.
Yeah, it's a pretty cool journey.
It is. We're exploring a discovery that honestly could really reshape how we study and maybe even treat the brain. We're talking about cryoRhodopsins.
Mm-hmm.
Now, if you're in neuroscience research, you definitely know about optogenetics,
Yeah.
using light to control neurons.
Absolutely. It's been revolutionary for understanding neural circuits.
Totally transform things. And the unsung heroes behind it are these microbial rhodopsins,
Yeah.
this huge family of light-sensitive proteins from microbes.
Right, like channel rhodopsins, which many people are familiar with. They're already staple tools in labs everywhere.
Exactly. So for this deep dive, we're zooming in on a really new group within that family, cryorhodopsins or cryers.
That's right. And they were found, interestingly, through bioinformatics first, basically screening genetic data.
So found in the computer before the test tube.
Kind of, yeah. And what's really striking is where they're found. These aren't your typical lab microbes. They come from organisms thriving in extreme cold. Think glaciers, permanent ice fields.
Wow. Like cryobacterium, things like that.
Precisely. Cryobacterium, subtrichal SSB, things that live on ice and snow surfaces.
Okay, and here's why this matters for, neurology and neuroscience. It seems their unique properties could give us a completely new set of tools for tinkering with brain activity.
That's the potential, yeah.
It's not just, oh, another protein. It's more like we've potentially found a new biological mechanism that could change how we tackle neurological conditions or even just understand basic brain function.
Exactly. So should we dive into what makes them so different mechanically?
Let's do it. Unpack these unique mechanics. What sets cryers apart?
Okay, so at the heart of it, structurally, is this really unusual arginine residue. It's buried deep within the protein.
Arginine, like an amino acid.
Yep. For instance, it's R57 in cryer R1, R58 in cryer R2. And what's weird is its location, it's really close to the cytoplasmic side, the inside face of the cell membrane.
And that's not typical.
Not for microbial rhodopsins, no. Even related ones like DSE rhodopsins don't have it positioned quite like that. And it's not just a structural oddity, it's functionally critical.
So this arginine is like a key feature. What about the overall structure? Do they work alone?
Well, they actually team up. They form pentamers.
It's 5 units too.
5 identical units, yeah. And we see this consistently, whether they're in detergent or lipid nanodiscs, or even when crystallized. It's a stable arrangement.
Okay.
And there's another piece. See, they have this quite long C terminus, the tail end of the protein. It seems to act like a cap over the center of this pentamer structure.
A cap. Does that do anything specific?
It's thought to contribute to their stability and maybe even those unusual kinetics we'll talk about.
Yeah.
Which, could be really useful if you want long-lasting control.
Right. Stability is key. Anything else structurally that makes them stand out?
There is, actually. It's the lack of internal water molecules right in the central region.
No water inside.
Well, very little, especially near the retinal binding pocket. That's the bit that actually absorbs the light. Most other microbial rhodopsins have water there, helping shuttle protons.
Oh, okay.
But in cryers, especially within about, say, 11 angstroms of the retinal shift base, that light-sensing core, it's notably dry. And this likely affects how they handle proton movement.
Okay, so unique structure, special arginine, no water inside. But the function, the light interaction, this is where it gets really wild, right? This slow dance idea.
That's probably their most striking functional feature. Their photocycle kinetics are incredibly slow.
How slow are we talking?
Okay, so when light hits them, they go into this intermediate state, the M state, which absorbs UV light. The amazing part is this M state can stick around for minutes.
Minutes, not milliseconds.
Minutes. Cryo R1, for example, takes 942 seconds to get back to its dark state at room temperature, around 20 Celsius.
That's over 15 minutes.
It is. And if you cool it down to 5 degrees Celsius, closer to their natural habitat, it stretches to 318 seconds.
That's almost an hour.
Nearly an hour.
It's orders of magnitude different from standard tools, like bacteriorhodopsin, BR. Its M state is gone in maybe 10 milliseconds.
Exactly. Milliseconds versus minutes. It's unprecedented for these types of non-bistable rhodopsins. Like you said earlier, a lingering glow versus a quick flash.
Okay. And does this UV sensitivity tie into their natural role?
It seems very likely. Because they're so UV sensitive, this M state can build up just from ambient light, like sunlight. So the idea is in their cold, high UV environments on glaciers, they might be acting as UV sensors.
Signaling danger from UV radiation, perhaps, makes sense.
Could be protecting the organism somehow.
But if they stay on for so long, how do you switch them off reliably for, say, an experiment or a potential therapy? Is there a quick reset button?
There is, thankfully. You can hit them with 420 nanometer LED light, basically, blue light. Researchers call it blue light quenching, or BLQ.
And that snaps them back quickly.
Yep. Rapidly reverts them to the dark state. And importantly, it does this without causing photo fatigue or photo bleaching. You can cycle them on and off repeatedly without breaking them.
Okay, that's crucial for practical use. No point having a switch you can only flip once.
Exactly. Robustness is key.
All right, so we've got this super slow UV-activated protein, resettable with blue light. What's happening under the hood, molecularly speaking? How does the light make it work, and what does it do?
So techniques like cryo-electron microscopy and X-ray crystallography have given us a really detailed picture. When it enters that long-lived M2 state, the retinal chromophore,
The light-absorbing part.
right, it changes shape. Specifically, it adopts a 13-cis conformation. And then that special arginine we talked about, R57, in CryoR1, it literally flips its position.
It moves.
It flips up toward the retinal shift base, the point where the retinal attaches. And this flip triggers a whole cascade.
And you mentioned this arginine flip does multiple things.
Yes, it's fascinating. It seems to play a triple role, all contributing to that super long on state.
Okay, what are they?
First, the flip lowers the pKa of the shift base. Basically makes it much harder for it to grab a proton back, locking it in the M state.
Got it. Keeps it activated.
Second, it physically gets in the way of another residue, a glutamic acid, preventing it from donating a proton back.
Blocking an internal proton source.
And third, by flipping up, this positively charged arginine creates a barrier. It blocks the path for protons trying to return to the shift base from the cytoplasm, the inside of the cell.
Like a molecular gatekeeper saying, nope, can't come back this way.
Pretty much. It's a really effective way to keep the state active for so long.
And what's the overall function that results from all this? Proton pumping.
Yes. The net result is inward proton pumping activity. CryoR1 definitely shows this. It pumps protons into the cell, but it needs white light with a UV component to get a steady current going.
Which again points to that UV sensor role in nature.
Exactly. Likely involved in maintaining pH balance or signaling in response to light conditions in its cold home.
And you mentioned it's even more complex, wavelength and voltage-dependent proton transfer.
Right. It's not just one simple action. Different colors of light seem to push protons in different brief directions.
How does that work?
Well, for example, pulses of 500 nanometer light green light seem to affect a state where a key internal group is deprotonated. This causes a brief positive transient current.
Meaning protons move outwards briefly.
Towards the extracellular side, yeah. But if you use 620 nanometer pulses red light, that affects a different internal state, one that's protonated, and that causes negative transients.
Protons moving inwards briefly.
Towards the cytoplasmic side. And get this, when you use that 420 nanometer blue light, the quenching light, on the M2 state.
The reset button.
Right. It doesn't just reset it silently. It causes strong negative transients, the biggest charge movement, actually. This is likely the shift base grabbing its proton back and the R57 arginine flipping back down.
Wow. So different colors can cause little shoves of protons in opposite directions even before the main pumping action or the reset. That's intricate.
It really is.
Yeah.
A sophisticated level of control.
Okay, so let's connect the dots. We have this incredibly sophisticated, slow-acting, light-controlled proton pump from icy microbes. Why is this exciting for neurology? What's the big deal for brain research?
The big deal really comes down to that minutes-long M state. It offers something researchers have wanted for a long time, the potential for sustained neuronal inhibition or modulation.
Sustained control, much longer than current tools.
Much longer. Most optogenetic tools give you effects for milliseconds, maybe seconds if you push it. We're talking about holding a neuron in a particular state, like inhibited, for minutes at a time. That's a huge leap.
Okay, I can see how that would be powerful. So, thinking about applications, maybe epilepsy, could you provide longer-lasting inhibition to calm down overactive circuits?
That's exactly the kind of hypothetical application people are thinking about. Instead of just brief suppression, maybe longer periods of stabilization.
Or for research, understanding neural circuits. If you can silence a specific group of neurons for several minutes, you could really see the impact on behavior or downstream activity in a way that's hard with just transient silencing.
Absolutely. It allows for probing circuit function on a timescale that's more relevant for many complex brain processes. Think about learning, memory consolidation, mood regulation. These things happen over minutes and hours, not milliseconds.
Right. So moving beyond just flicking switches on and off towards more tuning, like a biological dimmer switch you can leave on.
That's a good analogy, achieving precise, sustained tuning of neural networks, where transient control just doesn't cut it, either for research questions or potential therapeutic strategies.
And there was the other mind-bending possibility you touched on, bidirectional control, from just one protein.
Ah, yes, that stems from those opposing transient currents we saw with red and green light.
The little proton shoves in opposite directions.
Exactly. The fact that CryoR1 can do that, even transiently, with different colors, suggests the principle of bidirectional control using a single protein is possible.
Which would be a huge simplification, right? Don't current methods need two different proteins bolted together?
Often, yes. Complex tandem constructs are usually needed for robust bidirectional activation and inhibition. If cryers, perhaps after some engineering, could be developed to do both with different colors of light, well, that would be a major step forward in tool design. Simpler, potentially more targeted.
Okay, that sounds incredibly promising. But, science is never quite that easy. What are the hurdles? What needs to happen next to make these cryers practical tools?
Yeah, there's definitely work to be done. We're still in the early stages. A major focus for future research has to be engineering.
Tweaking the protein.
Exactly. We need to create variants, modified versions of cryers. One key goal is to get larger partial charge transfers from those red and green light pulses.
Make those transient currents strong enough to actually affect a neuron?
Precisely. Right now, they're likely too small on their own to reliably activate or silence a neuron. That's a critical limitation we need to overcome.
Makes sense. What else?
We also need to improve how well they get inserted into the plasma membrane of the target cells, like neurons. You need the protein in the right place to work effectively. So better membrane targeting is another engineering goal.
Okay, so make them stronger and make sure they get to the right spot. What about understanding how they work naturally? You mentioned they might have partners.
Right, that's another important avenue. Often, the genes for cryers are found alongside genes for other smaller proteins, putative cytoplasmic transducers.
Proteins they talk to inside the cell.
Potentially, yeah. These partners belong to a family called DUF7882. They look a bit like bacterial pH domains, and they also form pentamers, figuring out if cryers signal through these partners in response to UV light.
Could give us clues on how to harness that signaling.
Exactly. Or maybe even integrate cryer tools more cleverly into the cell's existing pathways. Understanding the natural system could inspire better engineered tools.
So lots of engineering and basic science still needed.
Definitely. It's promising, but still requires significant development.
Okay, so let's try and wrap this up. To summarize our deep dive today, CryoRhodopsins. They're this really fascinating new class of microbial rhodopsins found in extreme cold environments.
Distinguished by that unique buried arginine, and crucially, that incredibly long-lived M state lasting minutes, which is sensitive to UV light.
And the potential significance for optogenetics and neuroscience is huge. It offers this tantalizing prospect of prolonged neuronal inhibition or modulation. Could be a game changer for studying circuits or maybe even treating conditions like epilepsy.
Absolutely. And plus that hint of potential single protein bidirectional control, which would be a major simplification for optogenetic strategies.
But we have to acknowledge the limitations. Engineering is needed to boost their power and targeting.
Right. The fundamental mechanisms are exciting, the potential is there, but it's a pathway that needs further development before we see these in widespread use.
It really makes you wonder, though. Imagine the possibilities. If we could precisely control brain activity, not just for milliseconds, but for minutes at a time, what new insights into neural circuits, what potential treatments for really tough conditions might be unlocked by a protein that evolved just to sense UV light on ice?
It's a compelling thought, taking something nature evolved for one purpose and repurposing it for something entirely different in the brain. That's the power of this kind of research.
A fascinating area to watch. Thanks for joining us on the Deep Dive.